My presentation will start with a bit of introduction about protein retention in pigs, and I'll introduce a bit more about the impact of immune and stress response on protein utilization efficiency, and then I'll finish off with suggesting some of the available nutritional strategies that we can reduce the impact of immune and stress response on protein utilization efficiency. Nitrogen retention in pigs are affected by many different factors. For example, protein intake and body weight and physiological status and obligatory losses during nitrogen turnover and endogenous losses. And there are so many direct factors that are actually affecting nitrogen retention. It's not my intention to go through all this well-published research data. I've got all references in my paper, so if you want to be interested in, then you can go back to the paper. But the focus of this paper is just looking at the interaction between protein utilization efficiency and immune and stress response of pigs. If you look at the nitrogen retention in grower and finisher, if they take about 100 grams of nitrogen intake per day, then about 83% of nitrogen are digested and about 17% goes through the feces. And out of 87%, there are a lot of nitrogen oxidation occurring during the metabolism, which is comprised of about 51% of the nitrogen intake. And they go through the urine, and total loss are 68%. And the nitrogen retained in the pig, grower finisher pigs is around 32%. And there are large variation, as I said before, because there are large, a lot of different factors affecting it. And there are these are the reported variation in between different classes of pigs. But I just would like to highlight that this oxidation occurring during the metabolism of the nitrogen is affected by immune and stress response, and that's the factor that I'm going to go through in this presentation. I'll start with the immune response, and then I'll go and move on to the stress response and how they impacted the protein utilization efficiency. Pigs in the intestinal tract, they have this surveillance system. Somehow, in previous researchers say that it's as a subclinical infection. But in here, in villi and Peyer's patch, they have this dendritic cell, M cell, and CD4 cells have all ability to reach their arms outside of the lumen to sample what's in there, like antigens and pathogens, and they recognize their patterns and pass that information to the lymphoid tissues. And in the lymphoid tissue, if they found anything in the intestinal lumen, then they start producing immune molecules in the body to prepare possible open up or possible invasion of the pathogens, antigens into the body. And obviously, this is the energy and protein requirement process. Therefore, pigs in the dirty environment grow less efficiently and use nitrogen less efficiently. Then how much? If there was any quantification of this process, and this is a French study, looked at how's the pigs in the good sanitary condition and poor sanitary condition, and they found that there was about 5 to 7% reduction in feed intake and feed efficiency, and therefore their growth rate was about 10% lower. And there was a very similar report in poultry between germ-free and in the commercial farm. There was 5 to 7% reduction in growth rate. So we can, I can't really say definitely, but there is generally about 5 to 10% reduction when there's a subclinical infection. Then what happens when they get infected, inflamed? If they inflammate, I think previous researchers already introduced this very well, but inflammatory stress actually stimulates production of these two pro-inflammatory cytokines, interleukin-1 and TNF-alpha, which actually sending signals to the old bodies. They're sending signals to the liver and asking them to produce acute phase protein. And they initially use amino acid in the bloodstream, but if there is a short, then they ask, or they're sending signals to muscle to catabolize amino acid in the muscle and bring back to the liver. And then they also send the signals to the fat tissues to bring the fat or mobilize the fat to meet the requirement of the energy for this process. And this whole process actually changing dynamics of the partitioning of nutrition for muscle growth and fat growth and immune functions. This is the chicken study. I had to use it from Tony Edo's diagram. And they used a normal healthy broiler chickens and endotoxin challenged chickens. When they are healthy and they quantified how much nutrients are going for fat and muscle and lymphoid tissues, when they are healthy, about 70% of nutrients goes to fat and muscle growth, and there was only 1 or 2% used for lymphoid tissues. But when they challenged it with LPS, nutrients going to the lymphoid tissues are increased by 35% at the expense of fat and muscle growth. So if there is LPS challenge or endotoxin challenge, then we can say that there was about 30% of reduction in performance. And there was a very similar report in pigs. This is the result of the meta-analysis. They actually analyzed about 122 publications between 1960 to 2010. And this is the case of bacterial infection. And this is the control level. And these dots are the challenged pigs. In here, you can see that after 10 days of bacterial challenges, there was about 20% reduction in average daily feed intake. And if you see the average daily gain, average there was a 40% reduction in these pigs after 10 days of bacterial infection. So this number is very similar to what I showed before in chickens. And there's a 20% difference between intake and average daily gain reduction, which was explained by metabolic loss of nitrogen and energy during the responding to these immune challenges. Then what happens when pigs get stressed? And how they are actually affecting protein utilization efficiency in there. Stress actually stimulating this HPA axis called hypothalamus and pituitary and adrenal gland. And hypothalamus actually producing corticotropin releasing factor or corticotropin releasing hormone, which stimulate the pituitary and produce ACTH, and ACTH stimulate the adrenal gland. Adrenal gland produce glucocorticoids such as cortisol. All right? And this stress-induced glucocorticoids actually have direct and indirect effect for growth hormone release and for sex trade hormone release, and they actually increase visceral adiposity, but decrease muscle mass and bone mass as previously presented by Dr. Nick Gabula. And it's been tested in pigs in 2007. They just used a early weaning stress model, and they just weaned the pigs very early, like 16, 17 days. And then they stressed the pigs and then measured corticotropin releasing factor, CRF, in the colonic mucosa. And they found about 2.5-fold increase on CRF in the colonic mucosa. And also that stress actually associated it with increased paracellular permeability, about 2.5-fold again. So stress generally, increased stress hormone release, and this stress hormone actually increased intestinal permeability. And there's another study that's showing that increasing or exposing tissues to the stress hormone actually increase susceptibility to the live E. coli. And this study, they used the colon tissue from wiener pigs, and they exposed the colon tissue to luminal enterohemolytic E. coli. And that's the 0 level is control level. And there's another tissue they used. They exposed this tissue to ACTH, or expose the tissue with ACTH blocker plus ACTH. And they found that if tissue is exposed to this ACTH hormone, there was about 250% increase of bacterial attachment into the colonic walls. And this blocker didn't really bring that down back to the normal. So stress hormone increase permeability, but also increase number of bacteria actually can attach into the wall. This study showed a very good relationship or interaction between stress and disease in pigs. And what they did was they weaned pigs at 16 days, 18 days, and 20 days. So they gave more stress by weaning early and less stress by delayed weaning in here. And then they measured growth rate in control pigs and E. coli challenged pigs. If you challenge E. coli with early weaning stress, more severe stress, then there's a huge reduction in growth rate, while you challenge E. coli in the less stressed animals, there was a less reduction in growth rate. So stress and actually inflammation interact together. If I summarize all of these slides that I showed before into one mechanism, I think it will be a stress stimulating HPX axis, and they increase this stress hormone, and they increase epithelial permeability and increase susceptibility as well to secondary infection, and therefore they reduce growth rate. And this is the study conducted by Dr. Nick Gavlo's lab again, and they used the thermoneutral, they used these three different treatment, thermoneutral condition and heat stress condition, 37 degrees for 12 hours. And then they used another control, which is pair-fed thermoneutral, which is pigs put in 21 degrees, but they restrict-fed to heat stress counterpart to eliminate the compounding effect of reduced feed intake by heat stress. Sorry. And they measured plasma urea, which is an indicator of how nitrogen are efficiently used. Higher the number, less efficient. Higher the number, wasting more nitrogen through the urine. Okay? And they found that heat stress for 12 hours actually increasing plasma urea nitrogen by 2.5-fold. It's a huge increase, and huge amount of nitrogen are wasted during the metabolism or during the responses to this heat stress. And they found the increased plasma NEFA, which is an indicator of fat mobilization. They bring energy from the fat to address the required energy. And therefore, their body weight gain was significantly reduced during the heat stress. So far, I showed that how immune response and stress response can influence the nitrogen utilization in pigs. And I'm gonna suggest some of the nutritional strategies that we can minimize these responses, so that minimize reduction of reduction in nitrogen utilization efficiency. The first nutritional strategy is tryptophan and sulfur-amino acids. Tryptophan at immune challenges generally are metabolized to kynurenine through the enzyme called IDO, endolamine 2,3 dioxygenase. So if you challenge pigs with endotoxin or any bacteria, then IDO expression is increased, and more tryptophan are converted to kynurenine, which is used for free radical scavenging. And also this metabolites actually stimulate this transcription factor protein AHR, and this protein actually producing more anti-inflammatory cytokines in here and produce antimicrobial peptides to protect intestinal lumen. Okay, so generally, if there's an inflammatory challenge, then they use more tryptophan. So tryptophan became limiting for growth. And sulfur-amino acids is another one that actually using more severely when pigs are challenged with the immune system. Because sulfur-amino acids contains in lots of immune molecules contains a lot of sulfur-amino acids compared to muscles. So they use more sulfur-amino acids when the immune system is challenged to produce immune molecules, and therefore sulfur-amino acids start limiting utilization of other amino acids. There was a study conducted in Canada in 2010. What they did was they challenged grower finisher pigs with LPS, and they repeatedly challenged with that for about 6 weeks, and then they measured urinary excretion of sulfur and nitrogen. And they found that inflammation actually decreased sulfur excretion but increased nitrogen excretion, which means they used more sulfur but wasting a lot more nitrogen during the immune response. So we tested this theory in winner pigs at Murdoch University. This is the winner pig study. We used E. coli, live E. coli oral challenges, and then measured how much, what is the sulfur-amino acid requirement. In here, for growth, this is NRC requirement. But when pigs are challenged with E. coli, their requirement was increased by around 30%. And for gain to feed, there was also about 25% increase in requirement of sulfur amino acids. And this is the grow finisher study. And we measured this time protein deposition rate. And this black line is the saline injected pigs, and the red line is LPS injected pigs. As you can see, when pigs were healthy, there was not really increase any requirement of sulfur amino acids. But when pigs were challenged with the LPS, then the sulfur amino acid requirement actually increasing linearly by up to 25%. So it's very similar result from winner and grower. And then we tested these two amino acids together in winner pig models. And we challenged the pigs with the E. coli. And we challenged the pigs E. coli on day 5, 6, 7, and take the blood samples on day 5, day 8, and day 14. So we wanted to see response in day 5 before infection, and day 8, 24-hour infection, which is acute phase of response, and then day 14, which is 7 days after infection. So we wanted to see that response in the longer term, in an adaptive phase, if you like. And we measured APP index, which is acute phase protein index, calculated by this ratio, this ratio that we actually measured C-reactive protein and pigmap and ApoA1 acute phase protein, and calculate this APP index. So generally, higher the APP index, in the more severe response in inflammatory response. And as you can see, before infection, day 5, there was no inflammatory response. And acute phase response in day 8, 1 day after the infection, we found that in control pigs, which just fed normal NRC requirement of tryptophan and sulfur amino acids, found a huge increase in APP index, which means a huge increase in response to the inflammatory challenges. But feeding higher tryptophan and higher sulfur amino acids can suppress this inflammatory response. But in day 14, we found that tryptophan didn't really suppress it. So, but sulfur amino acid, was kept suppressing it up to day 7 after inflammation, which means tryptophan more working in very short term after inflammation in acute phase, but sulfur amino acid is working more longer term during this infection process. And from the same study, we see that increase in average daily gain by tryptophan, but there was a steady reduction in FCR for increased sulfur amino acid and increased tryptophan as well. And there was a sulfur amino acid and tryptophan interaction indicating use of both higher tryptophan and higher sulfur amino acid actually reduced further FCR in additive manners. Now, secondly, there was a tryptophan to large neutral amino acid ratio suggested, and increasing tryptophan to large neutral amino acid ratio in the diet can actually improve the response to stress. And large neutral amino acid is the sum of the valine, leucine, isoleucine, tyrosine, and phenylalanines. About 10% of tryptophan in the blood actually goes through this blood-brain barrier. And these tryptophan are converted to 5-hydroxytryptophan and then 5-hydroxytryptamine, which is 5-HT is serotonin. And they're actually sending negative signals to HPA axis, which I showed before, and therefore reduce production of CRF and ACT, HE, and glucocorticoids. So more tryptophan that goes through the blood-brain barrier, then there will be less response to stress. This is the blood-brain barrier. And this is brain side, and this is luminal side. As you can see, in brain side, there's lots of different amino acid transporters because they require to maintain homeostasis in the brain. If there's an excess or not required amino acid, they want to get rid of it quickly. So they have also different amino acid transporters. But in luminal side, there's only 2 transporters. And there's 1 transporter that actually is shared by tryptophan and large neutral amino acid. So if you got too much large neutral amino acid in the diet, then tryptophan transportation is reduced, and stress response will be more severe. So we want to increase tryptophan content in the diet, or we can reduce large neutral amino acid content in the diet to increase the ratio between tryptophan to large neutral amino acid. This is rat study that showed that increasing serum tryptophan actually linearly increased cortex tryptophan, and increased the cortex tryptophan also linearly increased hypothalamic 5-HTP, which is the precursor for serotonin. So if you increase rate of tryptophan transport to brain, you can actually reduce the response of the stress. It's in vivo pig study. They actually increased tryptophan ratio, tryptophan to lysine ratio in the diet, and measured concentration of plasma 5-hydroxytryptophan and salivary cortisol. And increased tryptophan, reduced cortisol, and increased serotonin. Okay, it's actually what should happen, or what should be happen. And in this study, they used different tryptophan, low, high, and used different large neutral amino acid ratio, 4.5 to 3.8% in the diet, sorry. And then they measured protein utilization efficiency. And as you can see, increasing tryptophan or reducing large neutral amino acid actually improved protein utilization efficiency. And this was translated to the huge increase in body weight gain. And exactly same study, they done about 3 years later. Not exactly the same, they modified a little bit here, but they found exactly the same response. So it actually can be duplicated, and the result was quite consistent. Then I'm going to introduce about some of the immune and stress modulators, which is my last part of the presentation. But most of them are very initial stage of research, and it's not in the stage of we can adopt in the commercial formulation. But these four strategies, like mast cell stabilizer, cortisol agonist, and cyclooxygenase inhibitor, and antioxidant, has a huge potential to improve protein utilization efficiency in pigs that are stressed by inflammation or general stress. Mast cell is the one of the immune cells in the body, and when they exposed to the stress hormone like CRF, they actually degranulating. They contain granule and degranulate, which is the enzyme to kill any pathogen coming through the body. But it is actually degranulating when pigs stressed. There's no enzyme, no pathogen invasion. But when they get stressed, they degranulate. They start damaging tissues. So this researchers used this mast cell stabilizer called cromolyn, sodium cromolyn. And they found that if you use sodium cromolyn, then you can actually suppress degranulation of mast cells after the exposure to CRF. And they also found that CRF increasing TNF alpha, but using mast cell stabilizer cromolyn actually can reduce the TRF production in the body. And they measured performance of pigs when used cromolyn in pigs. They used intraperitoneal injection of either saline or sodium cromolyn, and they found that they measured this intestinal permeability, and they found the cromolyn use actually reduced leaky gut syndrome, and they improved weight gain, and every daily gain plus gain to feed is also increased. But this is, there's only 2 papers published so far, so it's a very early stage of the research in this area. And another thing was a Texas Tech study published this year, and they used cortisol agonist, which is dexamethasone, and they injected intramuscularly day before weaning and 3 days after weaning, and they compared between unweaned suckling pigs and weaned suckling pigs, and weaned pigs with cortisol agonist in here. And they found that haptoglobin level is actually increasing when pigs were weaned. But if you use this cortisol agonist, you can actually depress haptoglobin production in the body. actually can decrease interleukin-1 beta cytokine gain, and they increase the body weight and increase the gain to feed ratio, which is a huge increase in here. If you can see, it was really reaching 0.9 in here. So this is another area, got huge potential to improve nitrogen utilization efficiency. And also there's an antioxidant that actually, or not antioxidant, in this case is the cyclooxygenase inhibitor, which is aspirin. Aspirin is actually reducing production of PGE2, which is the responsible molecule for neurological symptom of sickness. And if you use aspirin, then there was a huge improvement, not really improvement. In this case, we used the E. coli infection in here, but actually they can suppress waste of nitrogen during the metabolism or during the response to these immune challenges. And this also actually translated to growth performance and feed conversion ratio as well. And antioxidant is a huge thing because when pig's immune system is inflamed, they are producing a lot of reactive oxygen species, which damaging tissues. And if you use more vitamin E in here, after the E. coli infection, you can actually see vitamin E suppressing production of hepatoglobin in the body. And this was also tested in the commercial farm, and we found that higher dose of vitamin E in the commercial farm can improve feed conversion ratio for the first 4 weeks after weaning. So it's also quite a busy presentation, but the key point that I want to make in this presentation was exposure to inflammation challenges and environmental stresses. This is very common in commercial farm, and it actually can reduce efficiency of protein retention. So diversion of energy and amino acids to address such challenges resulted in a reduction in feed efficiency by 10 to 40%, which I showed before. And it's all depends on type and severity of inflammatory challenges. And then I suggest several nutritional strategies that can use to improve nitrogen utilization efficiency under these immune and stress challenges. I think I should finish here. Thank you very much for your attention.